Good morning, students. In this session we are going to discuss about mutations. What's are mutations, right? Translocations. Just now I told what it is. Translocations. These translocations are of three types. Translocations are of how many types? Three types. Translocations are of three types. What are they? First one is simple translocations. Second one is shift translocations. Third one is reciprocal translocations. Reciprocal translocations. Go for the first one. What it is, sir? Simple translocation. Simple translocation. Simple translocation. Now see here. Here I am taking, right, two chromosomes. Taking two chromosomes, right? A, B, C, D, E, F. This is one chromosome, sir. This is one chromosome, right? I am writing the next chromosome, right? G, H, I, J, I, J, K, L, right? Sir, this is another chromosome. These two are non-homologous chromosomes. What are these two? These two are homologous or non-homologous chromosomes. These two are non-homologous chromosomes. These two are non-homologous chromosomes. Sir, here what happens means a segment of chromosome. What, sir? A segment of chromosome from the ends gets removed, lost. See here. Sir, this is a segment of the chromosome lost from end of the chromosome. Okay. This segment comes and attaches to this chromosome. Comes and attaches to this chromosome. Therefore, after simple translocation, how it will be, sir? A, B, C, D, E, F, K, L. Okay, right? This is what, sir? Simple translocation. What happened here? Right? Segment of chromosome removed from the ends of the chromosome. That segment is attached to a non-homologous chromosome. This is simple translocation. Now go for the second one. What it is, sir? Shift translocation. What it is, sir? Shift translocation. Shift translocation. See here. Sir, here I am writing a chromosome, sir. A, B, C, D, E, F. I am writing another chromosome, sir. G, H, I, J, K, L. Sir, these two are non-homologous chromosomes. What are these two, sir? These two are non-homologous chromosomes. These two are non-homologous chromosomes. These two are non-homologous chromosomes. Sir, here what happens means, right, a segment of chromosome is lost. Segment of chromosome is lost. Sir, this segment of chromosome, is it lost, right? Is it lost? Is it lost from the ends of the chromosome? No. It is not lost from the end of the chromosome. Can be lost from any part of the chromosome but not from ends. Any part of the chromosome but not from ends. Okay, right? Sir, now this segment of chromosome, okay, comes and attaches to non-homologous chromosome. Comes and attaches to non-homologous chromosome at the end. No, it is not attached at the end. It is attached not at the end. Okay, can attach to any part of the chromosome but not at the end. Can attach to any part of the chromosome but not at the end. Therefore, now how the sequence will be, sir? After shift translocation, G, H, C, D, I, J, K, L. This is which translocation? Right, shift translocation. Therefore, segment of chromosome lost. That segment of chromosome is attached to non-homologous chromosome at any part other than ends. attached to non-homologous chromosome at any part other than ends. This is a shift translocation. Now go for reciprocal translocation. What it is, sir? Reciprocal translocation. Reciprocal translocation. Okay. What it is, sir? Reciprocal translocation. Now you see here, sir, A, B, C, D, E, F. This is one chromosome, right? G, H, I, J, K, L. This is another chromosome. Here what happens? See carefully. Sir, segment of chromosome last. What is last, sir? Segment of chromosome last. Segment of chromosome last. Sir, be careful. These two are non-homologous chromosomes. What are these two? These two, this and this, they are non-homologous chromosomes. They are non-homologous chromosomes. What happens means, this segment comes and attaches here. Right, this segment goes and attaches here. Okay, therefore reciprocal. Right, it means here exchange has taken place. Exchange of what? Segments of chromosomes. In between which chromosomes are? Non-homologous chromosomes. Therefore, exchange of segments of chromosomes between non-homologous chromosomes. Between what, sir? Non-homologous chromosomes. Therefore, after reciprocal translocation, how the situation will be, sir? How the situation will be? How they will be? A, B, C, D, right? K, L, yes or no? Right. And second one, G, H, I, J, E, F. Okay, therefore exchange of segments of chromosome between a non-homologous chromosome. This is called as what, sir? Reciprocal translocation. See carefully. Sir, in simple translocation, out of two non-homologous chromosomes, one chromosome lost a segment of chromosome, whereas one chromosome gained a segment of chromosome. Okay, therefore in one non-homologous chromosome, deletion, in another non-homologous chromosome, addition. See here, deletion in this chromosome, addition in this chromosome. Deletion in one, addition in another. But see this reciprocal translocation. Here what has happened, sir? Exchange has happened, that's it. No, right, deletion. Is there any deletion here? Sir, this part is okay, lost. Yes or no? But this part joined to this, and from here a part has came and joined here, no? Yes or no? Therefore, exchange has taken place here. Whereas in this case, right, deletion in one chromosome, addition in another. Right, deletion in one chromosome and addition in another chromosome. Okay, this is about what, sir? Translocations. How many types they are? 3 types. What are they? Simple translocation, shift translocations, and next one is reciprocal translocations. Right? With this, we completed translocations. Therefore, in translocations, right, in translocations, non-homologous chromosomes are involved. Yes or no? Which chromosomes? Non-homologous chromosomes are involved. Non-homologous chromosomes are involved. Okay, right? Now go for the next type of mutations, that is gene mutations. What it is, sir? Gene mutations. Sir, these gene mutations are also called as point mutations. What is another name for gene mutations, sir? Point mutations. Gene mutations also called as point mutations. Okay. What is the meaning of gene mutations? Change in single base pair, single one, right? Change in what, sir? Single base pair, BP. Change in single base pair of DNA. If it's DNA, change in a single base pair. Okay. If it's mRNA, if it is mRNA, then change comes in single nitrogen base. Therefore, change in what, sir? Single nitrogen base of mRNA. Single nitrogen base of mRNA. Sir, why here base pair, sir? Because DNA double stranded, no? Therefore, base pair, single base pair. mRNA single stranded, no? Therefore, change in one base pair. Right? Therefore, change in single base pair of DNA or change in one nitrogen base of mRNA is called as gene mutations or point mutations. Best example for gene mutations, is what's our sickle cell anemia. What is the example for, right, gene mutations or point mutations, sir? Sickle cell anemia is an example for gene mutations or point mutations. Okay, right? I think you got it in zoology. There is sickle cell anemia. Listen here. In first year botany, we read one statement. Sir, take a human hemoglobin. What it is, sir? Human hemoglobin, right? Sir, in this hemoglobin, this globin is a protein part. Yes or no? Globin. This globin is which part, sir? It's protein part. Sir, this globin protein, it contains 4 polypeptide chains. Remember, having how many polypeptide chains, sir? 4 polypeptide chains are 4 subunits. 4 polypeptide chains are 4 subunits. Sir, in these 4 subunits, in how many subunits? In these 4 subunits, 2 are alpha subunits and 2 are beta subunits. 2 alpha subunits, 2 beta subunits. That's why what statement we read in human hemoglobin, right? 4 polypeptide chains, 2 types of polypeptide chains. One is alpha, another is beta. Types are 2, but number is 4. Yes or no? Right? Now listen this statement. Sir, this sickle cell anemia, right? It is due to change in beta chain of globin protein in human hemoglobin. Therefore, in human hemoglobin, due to change in what's our beta polypeptide chain of globin protein, this sickle cell anemia has arisen. Therefore, sickle cell anemia, why, sir? It is due to, right, change in which subunit? Beta subunit, that is a beta polypeptide chain of globin protein. Of which protein, sir? Globin protein of human hemoglobin. Okay, sir, how this change has arisen? See here. Right? Now listen here. Sir, for example, right, this is a DNA molecule, sir. What is this one, sir? This is a DNA molecule, double stranded, no? Therefore, with 2 strands. Sir, this DNA, it has synthesized mRNA, sir. What is this one, sir? This is a mRNA. This is mRNA. Okay, right? Sir, this mRNA is made up of codons. mRNA is made up of what, sir? Codons. This is one codon. This is another codon. Like this mRNA is made up of many codons. mRNA is made up of what, sir? Many codons. mRNA is made up of many codons. Okay. Sir, from this mRNA, right, we are going to get a polypeptide chain. What we are going to get, sir? We are going to get a polypeptide chain. Sir, this polypeptide chain, it is made up of what, sir? Amino acids. This polypeptide chain is made up of what? Amino acids. This polypeptide chain is made up of amino acids. Okay, right? Therefore, from DNA we got mRNA. From this mRNA we got a polypeptide chain. Okay, now listen the statements. Sir, for example, this is the mRNA which synthesizes, okay, beta polypeptide chain. This is the mRNA which is synthesizing beta polypeptide chain. Okay, right. Sir, on this mRNA what are present, sir? Codons are present. What are present, sir? Codons are present. Here I am writing one codon. That is GAG. What is that codon, sir? GAG. What is that codon, sir? GAG. Okay, right. Sir, this GAG codon, right, GAG codon, it codes for glutamic acid. GAG codes for which amino acid, sir? Glutamic acid. It means now here what we should get, sir? Glutamic acid. Is this statement okay? Clear? Right? Therefore, see, this is mRNA. In this mRNA, right, GAG is one codon name. GAG codes for what, sir? GAG codes for glutamic acid. Therefore, when this mRNA synthesizes a polypeptide chain, that is beta polypeptide chain, right, here you are going to get which amino acid? Glutamic acid. Is this statement okay? Sir, in this beta polypeptide chain, this glutamic acid, it is at 6th position. I am speaking about glutamic acid which is present at which position, sir? 6th position. Now, due to mutations. Due to what? Mutations. Which mutations? Gene mutations or point mutations. Tell me. Gene mutations or point mutations means, change in how many nitrogen bases? One nitrogen base of mRNA. Here what happened means in mRNA, right, what is the codon? GAG. It has changed to GUG. Change has came in how many nucleotides? How many nitrogen bases? One nitrogen base. Should be GAG, but now it is what, sir? GUG. Sir, GAG codes for what, sir? Glutamic acid. But GUG codes for valine. GUG codes for what? Valine. 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 It means now instead of glutamic acid, what you will get? Valine. At which position? 6th position. Instead of glutamic acid, you will get valine at 6th position in what, sir? Beta polypeptide chain, which leads to sickle cell anemia. It means in a healthy person, right, in a healthy human being, if you see the hemoglobin, beta polypeptide chain having glutamic acid at 6th position. But due to mutations, a nitrogen base has changed. Okay, A replaced by U. So, GAG has became GUG, by which amino acid has changed. What is replaced by what? What you should get? Glutamic acid. But what we got? Valine. Therefore, glutamic acid is replaced by valine at which position? 6th position. 1st position, 2nd, 3rd, 4th, 5th, 6th. 6th position. Therefore, at 6th position, glutamic acid is replaced by valine. In which polypeptide chain? Beta polypeptide chain. Therefore, in beta polypeptide chain, at 6th position, glutamic acid replaced by valine, which results in sickle cell anemia. Results in what? Sickle cell anemia. Okay. Right? Here you should remember one important statement, what I told just now. What it is, sir? GAG. GAG, right, it codes for what, sir? Glutamic acid. Codes for which amino acid? Glutamic acid, amino acid. Okay, right. Sir, due to gene mutations, due to which mutations, sir? Gene mutations, due to gene mutations, due to gene mutations or point mutations, what happened, sir? What happened? Change in one nitrogen base, no? Right. A replaced by U, no? Okay, only change in one nitrogen base, gene mutation, point mutation, okay, right? But this GUG, it codes for what, sir? Codes for valine, codes for valine amino acid. It means glutamic acid replaced by valine, glutamic acid replaced by what, sir? Valine. At which position, sir? Sixth position. At which position, sir? Sixth position of what? Sixth position of a beta polypeptide chain, which results in sickle cell anemia. Which results in what, sir? Sickle cell anemia. Which results in sickle cell anemia. Is it okay? Right? Now, we are discussing what, sir? Gene mutations or point mutations. In this, sickle cell anemia is important. Therefore, you should be careful with sickle cell anemia, right? Now, go for the next statement. Sir, these gene mutations are of two types. Gene mutations are of how many types, sir? Two types. Gene mutations are of two types. Right? Gene mutations are of two types. Gene mutations are of two types. Right? What are the two types of gene mutations? Right? Number one, first one is substitutions. Substitutions. Okay. Second one is frame shift mutations. What is the second one? Frame shift mutations. This frame shift mutations is also called as a gibberish mutations. Gibberish. Gibberish mutations. Okay, right. Go for the first one. What is, sir? Substitutions. Name itself indicates. Substitutions. It means one nitrogen base is replaced by another nitrogen base. One nitrogen base replaced by another nitrogen base. Substitutions. Go for the first one. What is, sir? Substitutions. Substitutions. Okay, right? One nitrogen base is replaced by right another type of nitrogen base. Another type of what, sir? Nitrogen base. One type of nitrogen base is replaced by another type of nitrogen base. Sir, these substitutions are of two types. Substitutions are of how many types? Two types. What are they, sir? First one is transitions. What is the first one, sir? Transitions. Second one is transversions. What is the second one, sir? Transversions. Therefore, transitions, transversions. Go for the first one. What is, sir? Transitions. Sir, what is the meaning of transitions? Means purine replaced by a purine. Purine replaced by a purine. Purine replaced by a purine. Or pyrimidine replaced by a pyrimidine. What is, sir? Pyrimidine. Right. Replaced. Pyrimidine replaced by a pyrimidine. Okay, purine replaced by a purine or pyrimidine replaced by a pyrimidine. See here, for example, take adenine. Sir, in DNA, adenine is present, sir. If this adenine is replaced by guanine, adenine replaced by guanine, this is a purine. Purine is replaced by another purine, therefore comes under transitions. Okay, right. Sir, thiamine. Right? Sir, thiamine is a pyrimidine, sir. If thiamine is replaced by cytosine, thiamine replaced by cytosine. See, thiamine is a pyrimidine, sir. Cytosine is also a pyrimidine, sir. Therefore, pyrimidine replaced by pyrimidine comes under what, sir? Transitions. Therefore, purine replaced by a purine. It means one purine removed, another purine placed. One pyrimidine removed, another pyrimidine placed. Therefore, purine replaced by purine or pyrimidine replaced by pyrimidine, this comes under what, sir? Transitions. This comes under transitions. Okay. Now go for transversions. What is, sir? Transversions. Sir, in transversions, what happens means purine is replaced by pyrimidine or pyrimidine is replaced by purine. Therefore, purine replaced by what, sir? Purine replaced by pyrimidine. Purine replaced by pyrimidine or what, sir? Pyrimidine replaced by purine. Pyrimidine replaced by what, sir? Purine. Pyrimidine replaced by purine. Is this statement okay? Right? Purine replaced by purine or pyrimidine replaced by pyrimidine, it becomes transitions. Okay. Purine replaced by a pyrimidine or pyrimidine replaced by a purine, it becomes what, sir? Transversions. Therefore, transitions and transversions, these are two types of what, sir? Substitutions. These are two types of substitutions. Okay, right? Right? We are discussing, right, mutations. Now go for the second one. What it is? Frame shift mutations. What it is? Frame shift mutations, right? These frame shift mutations are either due to addition or deletions. Frame shift mutations are either due to what? Additions. Either due to what? Additions or deletions. Either due to additions or deletions. Okay. Frame shift mutations are either due to additions or due to deletions. Okay. We will get these frame shift mutations in detail in molecular biology. What is, sir? Molecular biology. Okay. Right. Now, go for the next topic. What it is? Mutagens. What it is, sir? Mutagens. Sir, mutagens means the agents which causes mutations. Agents which causes mutations are called as mutagens. Therefore, mutagens are nothing but what? Agents. Agents which causes what? Mutations. Agents which causes mutations are called as mutagens. Okay, these mutagens are of two types. Mutagens are of how many types? Two types. What are they? First one is physical mutagens. What is the first one? Physical mutagens. First one is physical mutagens. Okay. Second one is chemical mutagens. What is the second one? Chemical mutagens. Chemical mutagens. Okay, right. Go for the first one. What it is? Physical mutagens. What is the first one? Physical mutagens, right? Physical mutagens. Physical, right? Mutagens. Sir, physical mutagens, nothing but high energy radiations. What? High energy radiations. Should be careful. High energy radiations. Sir, they may be either ionizing radiations or non-ionizing radiations. Therefore, physical mutagens, nothing but what? high energy radiations. This high energy radiations which acts as physical mutagens are of how many types? Two types. What are they? Ionizing radiations. Second one is non-ionizing radiations. First one, ionizing. What is the first one? Ionizing radiations. Ionizing radiations. Sir, ionizing radiations example alpha, beta, gamma, X-rays. All these are ionizing radiations. What they are? Alpha, beta, gamma, X-rays. All these are ionizing radiations. Now go for the second one. What is non-ionizing radiations? Non-ionizing radiations. Non-ionizing radiations. Non-ionizing radiations. Okay. UV rays. Non-ionizing radiations, best example is what, sir? UV rays. Best example is UV rays. Okay. Now, see here. Sir, what is the example for non-ionizing radiation, sir? UV rays. UV rays, right? Now, you take a double-stranded DNA. What, sir? Double-stranded DNA. Double-stranded DNA. Okay, right? Sir, double-stranded DNA, two strands, no. Sir, these are the two strands. Okay, right? I am writing the sequence here. Random sequence I am writing A, T, G, C. Right, T, T, A. Random sequence I wrote there. Right? Now, what will be the sequence in the complementary strand, sir? T, right, A, C, G, A, T, T. This is the sequence in the complementary strand. Sir, if this double-stranded DNA is exposed to UV radiations or UV rays, what happens? If this double-stranded DNA is exposed to UV rays or UV radiations, what happens means, sir, this is thiamine, this is thiamine, sir. Sir, these two thiamines belongs to the same strand, yes or no? Sir, in between these two thiamines, a bond is formed by which thiamine dimer is formed. What is formed? Thiamine dimer is formed. Due to the formation of the thiamine dimer, right, this double-stranded DNA bends in this area. Bends in this area. Therefore, how it becomes, you see? Right, see here, sir, here what is there, sir? T is there. Here what is there, sir? T is there. Right, in between these two T's of the same strand or different strands, sir? Same strand. Okay, right, bonds are formed, right, by which a dimer is formed. Dimer, this is called as a, which dimer, sir? This is a thiamine dimer. Due to the formation of this thiamine dimer, right, the strand, right, the double-stranded DNA has undergone bending. Okay, right, due to this bending, what happens means mistakes takes place during replication. Mistakes takes place during replication. Mistakes takes place during replication. Therefore, when exposed to UV rays, what are formed? Thiamine dimers are formed. It means thiamines of the same strand, bonding is formed. Okay, due to the formation of thiamine dimer, right, the double-stranded DNA bends. Due to this bending, what happens? Leads to mistakes during replication process. Okay, this is about what's our physical mutagens, nothing but high energy radiations, which are of two types, ionizing and non-ionizing radiations. Now go for the second one. What it is? Chemical. Go for the second one. What it is? Chemical. Chemical mutagens. What they are? Chemical mutagens. Chemical mutagens, right? Chemical mutagens. Sir, these chemical mutagens are more harmful than, right, physical mutagens. Chemical mutagens, they are more harmful. Chemical mutagens are more harmful than what? Than physical mutagens. Physical mutagens. Why, sir? Because the action of a physical mutagen is localized. It is going to affect only one part of our body. Localized. Its action, its effect is localized. Whereas chemical mutagens, once enters into our body through blood circulatory system, distributes to different parts of our body. So, these chemical mutagens are more harmful than physical mutagens. Sir, through blood circulation, if a chemical mutagen reaches gonads, this chemical mutagens causes germinal mutations. What they cause us? Germinal mutations. Okay. Therefore, chemical mutagens. Which mutagens? Chemical mutagens. Chemical mutagens, right, spreads throughout the human body, by blood or through blood. Okay, right? And if reaches gonads, and if reaches what, sir? Gonads, causes, means chemical mutagens, causes what, sir? Causes germinal mutations. Causes which mutations, sir? Germinal mutations. Causes germinal mutations. Okay, right? Sir, we are having different types of chemical mutagens. Mustard gas is the first identified chemical mutagen. What is, sir? Mustard gas, first identified chemical mutagen. Okay, we are discussing chemical mutagens. Okay, go for types of chemical mutagens. Types of what, sir? Types of chemical mutagens. Types of chemical mutagens. Okay, right? Number one, first one, mustard gas. What it is, sir? Mustard gas. This is the first, right, identified chemical mutagen. First identified chemical mutagen. Who is the first identified chemical mutagen, sir? Mustard gas. Okay. Number two, second one is a nitrous acid. What it is? Nitrous. Okay. Nitrous acid. Sir, this nitrous acid, it is a deaminating agent. Nitrous acid, it is a deaminating agent. Deaminating agent means removes amino group from nitrogen bases. Okay. Therefore, nitrous acid is a what, sir? It is a deaminating agent. It acts as a deaminating agent. Sir, what these deaminating agents do, sir? Removes. Okay. Removes, right. Removes what? Amino group. Removes amino group. From where? From nitrogen bases. From where, sir? Nitrogen bases. From nitrogen bases. Okay. Right. For example, take adenine. What it is? Adenine. Sir, if adenine undergoes deamination, hypoxanthine is formed. What is formed? Hypoxanthine is formed. Sir, if you take guanine, what it is? Guanine. On deamination, this guanine forms xanthine. What it forms, sir? Xanthine. It forms xanthine. Okay, right? Sir, on deamination, cytosine. On deamination, cytosine forms a uracil. Cytosine has formed what? Uracil. Is it okay? Right? Now listen the statement carefully. Sir, take adenine, sir. Okay, adenine. Adenine pairs with what, sir? Thiamine. Yes or no? Adenine pairs with thiamine. It means this is AT pair. This is AT pair. What it is, sir? AT pair. Sir, due to deamination process, which process, sir? Deamination process, this what? Adenine has changed to what? Hypoxanthine. What has changed to what? Adenine has changed to hypoxanthine. Yes or no? What changed to what, sir? Adenine has changed to hypoxanthine. Yes or no? This adenine changed to what, sir? Hypoxanthine. Hypoxanthine. Sir, this hypoxanthine pairs with cytosine. Hypoxanthine pairs with what? Cytosine. It means it should be AT, no? It should be AT. But this AT pair has changed to what? AT pair has changed to hypoxanthine C pair. Hypoxanthine cytosine pair. Therefore, AT pair has changed to, that is hypoxanthine cytosine pair. Is it okay? Right? This is about which acid, sir? Nitrous acid. Which acid? It is a nitrous acid. We are discussing about types of chemical mutagens. We completed mustard gas and what, sir? Nitrous acid. Right? Now go for number 3. Go for the third one. Chemical mutagens, right? Right. Now go for the third one. What, sir? Alkylating agents. Which agents, sir? Alkylating agents. Number 3. Alkylating agents. Alkylating agents. Sir, these alkylating agents, right, they are used to remove purine. Used to remove what? Purine. Therefore, what they causes? They causes depurination. What they causes? Depurination. What is the meaning of depurination, sir? Removal of purine. Removal of what, sir? Purine. Removal of purine. Therefore, what is removed, sir? Purine is removed. For example, here I am taking a double-stranded DNA, sir. In this double-stranded DNA, here adenine is present, sir. What is present, sir? Adenine is present. Sir, adenine is a purine. Adenine is what, sir? It's a purine, right? Sir, due to this alkylating agents, what happened means this purine was removed, sir. What is removed, sir? This purine is removed. Sir, now there is a gap in this double-stranded DNA. This gap is filled either by using a purine or by using a pyrimidine. Is this statement okay? Alkylating agent, they causes depurination. It means remove the purine. If purine is removed, a gap is formed. GAP, a gap is formed. Sir, how this gap is filled? Either by adding purine or by adding pyrimidine. Actually, what is there? Actually, there is a purine. But now this purine is removed. This purine is removed. And what is placed there? Another purine is placed or another pyrimidine is placed. Okay, pyrimidine is placed. Okay, now you tell me, if purine replaced by purine, it becomes transition. If purine replaced by pyrimidine, it becomes transversion. Yes or no? Here purine is there, that purine is removed, gap is created. That gap is filled by using a purine. Purine replaced by purine, transition. Sir, purine is removed, gap is created. Gap is filled by adding pyrimidine. It means purine is replaced by pyrimidine, leads to what, sir? Transversions. Transversions. Okay, right? Alkylating agents. Which agents, sir? Alkylating agents. Best example is methane. Right? Okay. Methane. What it is, sir? Methane sulfonate. Methane sulfonate. Second one is ethyl methane sulfonate. Methyl methane sulfonate. Ethyl methane sulfonate. Okay, what they are? Methyl methane sulfonate. Okay, what is the second one, sir? Second one is ethyl methane sulfonate. Is it okay? Right? Therefore, methyl methane sulfonate, ethyl methane sulfonate. Right? Okay. Right? These two, right, they act as which agents are alkylating agents. Okay. Number 4. Go for the fourth type of chemical mutagens, base analogues. What, sir? Base analogues. Base analogues. Sir, these base analogues, they resembles nitrogen bases. Base analogues resembles nitrogen bases. Resembles what, sir? Nitrogen bases. Resembles nitrogen bases. But what is the difference means? Sir, these base analogues, they are resembling the nitrogen base, but a normal nitrogen base, it pairs with what? A base analog won't pair with that. Is this statement okay? Right. Sir, a normal nitrogen base with what it is pairing, a base analog won't pair with that nitrogen base. A base analog won't pair with that nitrogen base. Okay. Therefore, base analogues, they resembles what, sir? They resembles nitrogen bases. They resembles what, sir? Nitrogen bases. For example, you take 5-bromo-uracil. 5-bromo-uracil is a base analog of thiamine. 5-bromo-uracil is a base analog of what, sir? Thiamine. It's a base analog of thiamine. Okay, right? Therefore, this is about what, sir? Base analogues. Now go for the next one. What, sir? Other chemical mutagens. Other what? Chemical mutagens. Other chemical mutagens. Other chemical mutagens. Other chemical mutagens. You can take a DDT. Right. Even you can take colchicine. In first year we read. Yes or no? Sir, this colchicine is a mitotic poison. Colchicine is a mitotic poison. Yes or no? Sir, this colchicine is extracted from Colchicum autumnales. Yes or no? Meadow saffron. Right? This colchicine is a chemical mutagen. It's a mitotic poison extracted from, right, Colchicum autumnales. There is a meadow saffron. Sir, this meadow saffron belongs to which family, sir? Liliaceae family. Like this, right, many other chemical mutagens are there. Right, we discussed only two here, DDT and what, sir? Colchicine. Okay, right, this is concerned to what, sir? Chemical mutagens. Therefore, mutagens two types: physical mutagens, chemical mutagens. Physical mutagens, radiations, either ionizing radiations, non-ionizing radiations. Chemical mutagens, right, first identified chemical mutagen is what it is? First identified chemical mutagen, mustard gas. Right, and second one is what is the second one, sir? Nitrous acid. Third one is alkylating agents, and fourth one is base analogues, and next one is DDT and colchicine. Okay, right? Thank you.